Hello students, I Anil Kumar. Today I am going to start the second lesson of the plus 2 biology under the title Sexual Reproduction in Flowering Plants. As you know that every flowering plant can show the reproduction, and in the reproduction there the three different steps will be there. The first step is the pre-fertilization, second step is fertilization, and third step is the post fertilization. Clear? So just we going to start the pre-fertilization. In the pre-fertilization, on the flower there are the four different parts. The first part, that one, that name as the corolla, that name as the corolla. The corolla is the colored part of the flower. It also named as the petals. Okay. The second part is the green one, named as the calyx or the sepals. That name as the sepals. S-E-P-A-L. Sepals. Okay. The third part is the female part. That is the gynoecium. Gynoecium, or say the pistil, consists of the three major parts. The first part is the stigma, then style, and then the ovary. The fourth one is the male part. Okay, that consists of stamens. Okay, when we just cut it and showing the detailed structure, so it look like that one. That is the bithecus. It contains the pollen grains or the Microsporangium. While if we cut the ovary part, so it look like the that one. So it contains the number of ovules. That depend upon the species to species. It is a single ovule or the multi ovulate is there, right? So in the pre-fertilization, so in a flower, first we are going to study the flower. Okay, so in a flower, what we are going to study in the male part, that is the stamen, then how the anther is formed, then the microsporogenesis, formation of the microspores, and then the strategy of the pollen grains. While in the female we are going to study the structure of pistil, then structure and of ovule, then the megasporogenesis, at last is the embryo sac. Clear students? So let's be going to start our chapter. So like that we already studied the flower, because there will be the one primordial cell. Always remember, before the formation of anything here in the plant, there will be a need of the primordial cell. So like the primordia, here is the fluoroprimordia is present. Fluoroprimordia change into the floral bud, and floral bud is modified into the flower after getting the change due to the hormonal and the some structural change. So it flower is the modification of the stem part. Clear students? How the stem can be modified into the flower? It is only by due to the structural change and the hormonal change. As you know, the phytohormones play the very important role here, right beta? Then come on the male part. So male part is named as the anther. Okay, so in the anther part, if we going to cut that anther from here and here like that one, so it look like that. So that is the four compartment are there. One, that is the second part, and here from this side 3, and that is the 4. Okay, there is the 4 part are there, 4 compartment there. That 4 parts are there. That means plant will become as the dicotyledon. If a monocotyledon is there, so that means the one side is like that one, another side is also that one. Okay, that is the monocotyledon, but in that case is for the dicotyledon. Okay, that part is consist of, that is the filament, that part. Okay, and the upper part is named as the anther. Filament should be attached with the thalamus part, or say the sometime with the petals. Okay, when it can be fused in nature. Okay, then structure of the anther. How to describe the structure of anther? So as you know that it is a bithecus. That is a bithecus. The two lobe is there. Okay, bilobe or dithecus. A longitudinal groove separate the theca. That should be separate the theca layer. In the cross section, anther has the consists of the 4 microsporangia, 2 in each side. These are the microsporangia, 2 in each side. Okay, a microsporangium, अकेला microsporangium की बात करें तो ऐसा होगा. That is a microsporangium. It consists of the microspore mother cell first of all. Clear? That is a microspore mother cell. Clear, students? Then come on the microsporangium. Microsporangium is, or the structure of the anther consists of the 4 different layers. The outermost layer is the epidermis, which help in the protective in nature. Second layer is named as the endothecium. Third layer is the middle layers. It consists of 2 of the 3 layers. And the much more important is the tapetum. Tapetum is the very important as per examination point of view, because tapetum can be provide the nourishment. It provides the nourishment for the developing grains, while the epidermis can be help in the dehiscence protection. Okay, endothecium and the middle layer is also going to that one. The same is the protective in nature. Clear? You just going to study the structure of that one. Then this is the cut part of the microsporangium, and that is microsporangia. If we going to check in detail, so like that one. So that is the first layer, epidermis. That is the outermost layer. After that one, it should be covered by the endothecium, and the covered by the another layers are the 2 or the 3 layers of the middle layer are present. After that one is the microspore mother cell. That layer, that abbreviation is the MMC. Microspore mother cell. That's these cells are the differentiated from the other cell only due to the presence of the dense cytoplasm as well as the nucleus. These are the nucleus. Okay, that is the structure differ from other cell is there. Why? Because nucleus will be present here. Clear, student? This is the diagram showing the dehiscence. When the 4 sporangium are there, here is the 4 sporangium, both the side, they are merged together. They are merged together like that one. So it look like here. Okay, this is the dehiscence stage, and then that is the dehiscence of the pollen grain take place. Clear? When it should be dehiscence, so what happened? So simply first define the definition of microsporogenesis. Microspore means the male. Okay, genesis is the formation. So here is the microspore mother cell can be formed the tetrad. Here is the tetrad, and they can be developed all the 4 microspore, or they all are capable. They are all are capable. Clear? To form the pollen grain, right? While in the case of female, only one is functional. That is the major difference. In male, all the 4 are functional, while in the female, only one is functional. Okay, beta? So let's again we just define the definition. The process of formation of the microspore from the microspore mother cell through the meiotic division is called as microsporogenesis. So it form equally divided into the first of all the 2, then 2 divided into the 4, and 4 all are the functional in nature. On maturation, the dehydration of the anther, spore dissociate and develop into the pollen grain. Okay, and pollen grain release with the dehiscence of the anther. Clear? Now we going to study the pollen grain structures. In the pollen grain structure, it is spherical in nature. It is a spherical in nature. It has the two layer. It has two layers. The outermost layer named as the exine. Exine layer should be fully covered the it fully covered the pollen grain except the few area. These are the area where exine will be absent. Why? Because that is the area where the intine can produce the pollen tube. Okay, or it is the area where there is a one pigment, there is a one chemical present on the exine that name as the sporopollenin. That is a substance who resists the pollen grain from the organic matter, from the high temperature, acid, alkali. That is the only reason that they become as the good fossil. Even for the more than 100 of years they can be survive only due to presence of that substance sporopollenin. Okay, and after the exine layer, that is the second layer is named as the intine. Okay, in the intine, that is a very thin layer continuously made up of the cellulose and the pectin. So sometime examiner will ask, write the difference between exine and the intine in case of the pollen grain. So major difference is their chemical composition. Exine layer will be formed with the help of sporopollenin, while the intine can be made by the cellulose and the pectin. Exine will be the discontinue covering around a pollen grain, while intine will be the complete layer, the complete covering. Okay, the third one is the major that intine can produce the pollen tube while the exine can't be. Okay, there will be the outgrowth occur in the case of the exine. There will be the some rupture or the some thorns like or the rough structure will be present, while in the case of intine it does not. Right, beta? So that is the major things we describe one by one here. Now let's be going to study the structure of the pollen grain in detail. If we going to study of the pollen grains, so there are the two type of cell. One named as the vegetative cell, which is the larger, okay, larger as compared to the generative cell. Clear? And vegetative contain the food reserve material also, okay, but having the irregular nucleus, while in the case of the generative cell it should be the regular spherical, or it will be the float in the vegetative cell. Clear? In the mostly 60% species, pollen grains set in the 2 cell stage, while the 40% is there set in the 3 cell stage. Clear? Or then that will be the mitotically divide and to form the 2 male gamete. Clear, students? That is the 2 male gamete form. Now we going to start the economic importance. It is not much important as per the examination point of view, but something is there. So please learn very well accordingly your need. So as you know that its outer nature or outer layer of the exine of the pollen grain is the exine, and exine has the some spine type of nature is there, hard nature is there. Okay, when you sometime you visit in the garden, so there will the rose are there, hibiscus, China rose is there. When you touch the flower, so the some powdery like substance just take on your finger, so that is the pollen grains. Okay, it causes the allergy when we respire, when we going to breathe, so some pollen grains we can inhale. Okay, so during the inhaling, so it's going to some problem in the respiratory tract. Okay, so that's why it causes the allergic problem. So like the asthma and bronchitis, it can be caused by that one. And the pollen grains of the Parthenium, you know the Congress grass, it show the allergy also, it causes the itching in the body. But pollen grains are the rich in nutrients, rich in the protein especially, so they act as the food supplement. Pollen consumption increase the performance, yeah, that means it increase the efficiency of the body in the athletics or the in the horse who participated in the races. Okay, This after setting, their viability depend upon the temperature and the humidity. As you know that the 30 minutes will be the time for their viability in case of the wheat and the rice. While in the Rosaceae family, leguminous and the Solanaceae family, they remain viable for the month. Remember that point, beta. Then that is very important. Okay. Why? Because like the our germplasm or like the other things, we store the sperm, so like these other things also going to store. So what is the method we can use normally for the preservation? So that preservation is named as the cryopreservation. Remember that one, cryopreservation. Cryo mean that is the low temperature or at as the coolest area. Okay, so then we use the here one gas is the name of the nitrogen at 196 degrees Celsius we going to preserve. Okay, that name as the cryopreservation. So remember that point. Okay, clear students? So if you have any doubt related to that chapter, especially in that topic of the pre-fertilization change, so you just drop the one message on the WhatsApp number 96712000638. For any query at any time, you will be just drop the one message or drop the message to add in my group. Also, you also email me on Anil Kumar Ghangas, ANILKUMARGHANGUS @gmail.com for any query in the biology. In the later session, we going to discuss the fertilization with the Microsporogenesis. Right students? Thank you for watching the video.